A very good morning to all of you. Coming to the last bit of systemic bacteriology, this is a chapter on mycoplasma. Till now, we have dealt with all the gram-negative organisms. So, mycoplasma also comes under this category of gram-negative organisms. Mycoplasmas, as we all know, are cell wall deficient forms. They are the smallest known organisms. Because of their cell wall deficient forms, they usually cannot take up the gram stain. So classically, we cannot describe them as gram positive or gram negative. They can take up the Giemsa stain much better than they can take up the gram stain. So under this chapter, mycoplasmas, we will be dealing about the morphology of mycoplasma, then the different infections which are caused by them, how you can make a diagnosis of the infections caused by mycoplasmas, and lastly, how do we treat the conditions caused by mycoplasma. A little bit on the introduction to mycoplasmas. It were these men, Nocard and Roux, who first identified this organism from cases of bovine pneumonia. Bovine meaning cattle. So this organism was first isolated from the sample of the infected cattle, and they gave the name as pleuropneumonia like organism, which in short is referred to as PPLO. Later on, it was renamed as mycoplasma because of its morphological appearance to the fungus, which has branching filaments, that is a mycose, and since they have no cell wall, they have a high rate of plasticity, therefore they can take up any shape. So therefore the name plasma. Mycoplasmas are a group of bacteria that are devoid of cell wall, so therefore are highly pleomorphic with no fixed shape and size. They have certain characteristics such as the cell wall precursors itself are absent in mycoplasma, which in contrast to the L forms, they lose their cell wall over time, but they do have their cell wall precursors. But in mycoplasma, the precursors for making the cell wall itself is totally absent. So what is there instead of the cell wall? There is a trilaminar unit membrane which contains or which is made up of sterols, that is cholesterol and lipid. So the high plasticity makes them to pass easily through bacterial filters, for which they were mistaken to be viruses. But then later on, they realized there are certain features for which the mycoplasmas do not belong to the viruses, but they are bacteria. Coming to the classification of mycoplasmas, they belong to the class of mollicutes, which means having soft skin, the order of Mycoplasmataceae, and the family Mycoplasmataceae, which has two genera, that is Mycoplasma and Ureaplasma. The other families under the order Mycoplasmataceae is Ecolplasmataceae, Spiroplasmataceae, and Aneroplasmataceae. Each of them have different characteristic features, and they are not known to cause human infections. In this slide, we will be seeing the different species of Mycoplasma and Ureaplasma which are important for us, and the different infections which are caused by them. In this table, we can see the different species and each species causing the respective infection. First and the most important one is Mycoplasma pneumoniae, which is known to cause infection of the respiratory tract, both upper as well as the lower respiratory tract. It is also known to cause primary atypical pneumonia, which I will be telling you in the following slides. Mycoplasma hominis is another species which is known to cause pelvic inflammatory diseases. Mycoplasma fermentans, which is known to cause opportunistic infections in HIV individuals. Then we have another species, that is Mycoplasma pyrum, which again is known to cause septicemia among the HIV individuals. Ureaplasma ureolyticum, it is known to cause pelvic inflammatory diseases, and particularly in the females, it is causing vaginitis and cervicitis. Coming to the morphology of mycoplasma, as already mentioned, it is the smallest known microorganisms, measuring only 150 to 250 nanometer in size. They are devoid of cell wall and they are made up of only a trilaminar unit membrane made up of sterols. Because of lack of cell wall, they are highly pleomorphic. They can take up different morphologies. They can either appear as granules and filaments of different sizes. Even among the granules, they have classical shapes that they can take up. Sometimes they can appear cocoid, and the same cocoid can just balloon out and they'll appear like a blown up balloon. They take up the shape of a disc, a ring, and a star form. They do not have any fimbria and flagella. Since they do not have a cell wall, they are poorly stained by Gram stain and they take up the genes are stained better. Coming to the antigenic properties of mycoplasma, they have the surface antigens which is made up of glycolipids and proteins. What is the nature of these glycolipids? They show cross reaction with the human tissues with similar antigenic structure to that of the neurons in the human brain. Therefore, the mycoplasmas are also known to cause neuronal damage. Coming to the protein antigens, they are the major antigenic structures of mycoplasma which is known to cause the respiratory infections, and there is only one major protein which is known as the P1 which helps in the attachment to the receptor surface. And as well as they are more antigenic, therefore antibodies are developed which can easily be identified by ELISA. Coming to the pathogenesis of mycoplasma, the host is always the humans. Mycoplasmas are not known to cause infections among the animals. There are species which are classified under the different families, which I already showed you in the classification slide. The mode of transmission for the infection is through the nasal secretions and sometimes through inhalation of the aerolized droplets. Mycoplasmal infections are found to have a worldwide distribution. The infection is found highest among the school-going children and it has one virulence factor, that is the P1 antigen. Coming to this different sequence of events, what takes place? Firstly, it is the P1 antigen which adheres tightly to the epithelial cells in the respiratory tract. They cause direct damage to the epithelial cells along with the cilia, and this interferes with the normal functioning of the upper respiratory tract. Now, the infection slowly trickles down from the upper respiratory, moving down into the lower respiratory tract, which gets infected with the microbes, and there is mechanical irritation, which ultimately leads to the persistent cough, which is very typical of Mycoplasma pneumoniae infections. Coming to the different manifestations of Mycoplasma pneumoniae. It is known to cause the respiratory infections, the upper as well as the lower respiratory. In the upper respiratory tract infection, the patient usually presents with fever, malaise, headache, and there is non-productive cough. Coming to the lower respiratory tract infection, the patient presents with tracheobronchitis and bronchopneumonia. There is a condition which is caused by Mycoplasma pneumonia, which is the primary atypical pneumonia, which is known as walking pneumonia or working pneumonia. Okay, I am sure all of you all have seen cases of pneumonia wherein the patients are very toxic with very high fever, they are totally bedridden and with persistent cough. But that is not the case with primary atypical pneumonia. Therefore, the name atypical. The typical symptoms of pneumonia are usually absent. Okay. The incubation period is for about 2 to 3 weeks. The patient will present with cough. The patient appears totally normal. He gets up in the morning, he does his usual routine work and he goes to office. So, he does not find that there is something, he has an illness. Apart from the cough, he is totally fine. But because of the persistent cough, he is made to go to a clinician. On examination, they find that the pharynx is edematous. Then when they ask for a chest x-ray, there we can find the bronchopneumonia patches. So, there is a disparity between the presenting complaints and the radiological evidences. So, therefore, it is known as atypical pneumonia. And the beauty of this is it is self-limiting. So atypical pneumonia, the patient does not have fever, has no chills, there is no pleuritic pain. Also, there is no expectoration of the sputum, which are classical symptoms of pneumonia. Okay, a typical pneumonia. But Mycoplasma pneumoniae causes atypical pneumonia, where all these symptoms are totally absent. So, which are the other organisms which are known to cause atypical pneumonia? Apart from Mycoplasma pneumoniae, we have Chlamydia pneumoniae, Legionella pneumophila. And sometimes we fail to isolate the organism from the sputum sample. Atypical pneumonia usually asks for 3 marks. Coming to the laboratory diagnosis for Mycoplasma pneumoniae infections. First, what is a specimen that we need to collect in such kind of conditions? Ideally, we can collect a throat swab. Then we can go for also the bronchial washings, and if we don't get good samples, then we go for the sputum sample. Microscopy is of no value, as already told, because they lack the cell wall, they do not take up the gram stain. So we can do a direct antigenic testing. So the methods that are usually employed is a direct immunofluorescent technique and also immunoblotting. We can also grow this Mycoplasma organism. So they have selective media, which is known as a PPLO broth and PPLO agar. As we all know, broth means liquid media. So what does this PPLO broth? PPLO stands for pleuropneumonia-like organisms. So PPLO broth, it contains 20% horse serum, 20% yeast extract, 10%. It also contains glucose, and the indicator that is added is phenol red. So the sample of choice is inoculated into this liquid media, and the growth is observed for over 1 to 3 weeks. And we can see that there is a change in color of the media. It changes from red to yellow color because there is change in the pH. And we also should look for the turbidity in the liquid broth. If we add agar to the PPLO broth, it becomes PPLO agar. So on the PPLO agar, the sample is inoculated. And then following incubation, we find the classical or typical fried egg appearance colonies of Mycoplasma pneumoniae. So what is this fried egg colony? I'll be telling you in the next slide. And these are identified by what is called as a Dyne's test, wherein we use a Dyne's stain, pour it on the plate, and we look for the appearance of the colonies under the microscope. We also do a heme adsorption test, wherein the guinea pig RBCs are added, and if the Mycoplasma organism is present, it absorbs the RBCs onto its surface. And we can also do a tetrazolium test and look for the color change. If all these tests come positive, then we are definitely dealing with a Mycoplasma pneumoniae organism. So this is a typical fried egg colony which I was telling you about, and this is after adding the Dyne's stain, wherein you can see the central portion is taking up the deep blue color, and as it moves away from the center, the periphery is appearing light blue in color. The incubation for the plate is about 2 to 6 days. The size of each colony measures from 10 to 600 microns in size, and it is mentioned as fried egg appearance, okay, or also known as a biphasic fried egg appearance, because it has 2 faces there. The central opaque granular area surrounded by a flat translucent peripheral zone. After doing the culture, we can also do certain serological tests which can give us more information regarding the infection. So it is classified into 2 types: the specific and sensitive tests and the non-specific tests. Why it is known as specific is because we are using the Mycoplasma antigen itself to identify it. Okay, so non-specific means we are not going to use a mycoplasma antigens, but we are going to use a substitute of the mycoplasma, which is known as heterophile mycoplasma antigens. So what are the different specific and sensitive tests? We can do an indirect immunofluorescent test, heme agglutination inhibition test, and also ELISA to demonstrate the different specific antibodies using the specific mycoplasmal antigens. Therefore, it is known as specific, and these tests are more sensitive. Therefore, they are known as specific sensitive tests. Coming to the non-specific tests, they are called so because we are not using the mycoplasma antigens, but we are using a substitute for that, which is known as the heterophile mycoplasma antigens. So, what are the two tests? First one, we have the streptococcal Mg test, and the second one is the cold agglutination test. In the streptococcal Mg test, we are using the streptococcus as our antigen to detect the level of the antibodies. So what do we do in this test? This test is done by mixing serial dilutions of the patient's unheated serum under heat-killed suspension of the streptococcal Mg strain. We incubate this and we observe at 37 degrees Celsius, and we look for the titers the next day. Similarly, how we do for a Vidal test, this is the same procedure. A titer of 1:20 is suggestive of Mycoplasma pneumoniae infection. Okay. Coming to the cold agglutination test, serial dilutions. In this test, we are going to use human blood group O RBCs as our antigen to detect the level of the antibodies. So, serial dilutions of the patient's serum is mixed with equal volume of human blood group O RBCs, and the clumping is observed at 4 degrees Celsius overnight. The next day, when we look for the titer of the antibodies, a titer of 1:32 or above is suggestive of Mycoplasma pneumoniae infections. So in both these tests, you can see that we are not using mycoplasma antigens, but we are using a substitute for them to detect the level of antibodies against the mycoplasma. So these are known as the heterophilic agglutination tests. Coming to the treatment for Mycoplasma pneumoniae infections, the drug of choice is doxycycline, which is given in a dose of 100 mg twice a day. If the patient is found to not react to doxycycline, then they give erythromycin, which is given 500 mg 3 times a day. That finishes the topic on Mycoplasma pneumoniae. Coming to the genital Mycoplasma infections. As we saw in the first slide, the table wherein it had different species. So among them, there are certain species which are particularly known to cause genital infections. So they are Mycoplasma hominis, which again particularly affects the females, causing pelvic inflammatory diseases, salpingitis, and also commonly causes septic abortions. Mycoplasma genitalis, it is known to cause NGU, which is non-gonococcal urethritis, which has already been completed in the chapter Neisseria gonorrhoeae. This species is also known to cause pelvic inflammatory diseases. Coming to what is this Ureaplasma ureolyticum. This also belongs to the same family, Mycoplasmataceae. Ureaplasma ureolyticum, this is a genital pathogen. It causes sexually transmitted diseases. It is also known to cause non-gonococcal urethritis, chorioamnionitis, vaginitis, and cervicitis. It is diagnosed by culture and PCR. Why it is called as Ureaplasma ureolyticum is because it requires the supplementation with urea for their growth. Unlike Mycoplasma, which does not require urea, this organism requires urea. Thank you.